Okay, so we covered our innate defense, or non-specific defenses, which is our first and second lines. Now we're going to look at our acquired, our specific defenses. However, we need to have a little bit of background information before we go into more detail. So now here's our problem. What happens if a pathogen gets past your first and your second line of defense, and now it's circulating throughout your body, or hiding or living within a cell, right? This is where we have our third line of defense. And our third line of defense utilizes and works with our circulatory system and our lymphatic system. So I know in your life you probably have learned about the circulatory system with blood vessels at some point or another. But one thing that we don't really teach about too often is the lymphatic system. Kind of a cool thing where you have blood vessels, arteries, capillaries, and veins that your blood circulates through. You also have vessels called lymphatic vessels that lymph fluid circulates through. So if I were to zoom in, I can see here in this capillary bed, there's also lymphatic vessels mixed in with the capillaries. And so when we talk about lymphatic fluid, think about your blood and your lymphatic fluid. Your lymphatic fluid is like a clear fluid. So it doesn't have red blood cells. It's basically collecting any water or amino acids, like salts, little things that seep out of your capillaries into your tissues. Your lymphatic fluid collects back. So your lymphatic channels role is to collect fluid in your body and then put that liquid back into your bloodstream. So if we look at this picture here, you can see the lymphatic channels will actually drop the fluid back into your blood, so that way your blood volume stays the same. So you don't lose water. It gets put back into your blood vessels. So here your blood and your lymph are circulating through your body. And so what we're going to focus on and talk about are lymph nodes. Okay? So when we look at this, here I can see my lymphatic system, or a lymphatic system. And here's more looking at the lymphatic channels. And then here you can kind of see how a lymph node has both lymph fluid flowing through it and capillaries as well. So when we look at the lymphatic system's role in the immune system, here we have two types of lymphocytes. Okay, so we have B cells that are made in the bone marrow and then will finish development in the bone marrow. And then we have T cells. T cells are going to be made as well in the bone marrow, but will actually travel to the thymus, and that's where they'll finish development. So our B and T cells are both made in bone marrow. B cells stay there and mature, and T cells travel to the thymus and mature there. All right, box 6. And so when we look at the other aspects of our lymphatic system, we see here we have lymph nodes in different places throughout our bodies. And so when we look at lymph nodes and what their role is, we find that macrophages and dendritic cells and lymphocytes, they live here and they attack microbes circulating in the lymphatic fluid. So if I just go back real quick, oh my gosh, if I go back to this, you can see here, if you have macrophages and lymphocytes and immune system cells living in this lymph node, that's pretty awesome for when the blood or lymph carrying pathogens circulate through here. So here in the lymph nodes you have an area where macrophages, which are our big eaters, are going to live. And you might also have B cells and T cells. The lymphocytes may live here as well. And so if you have a pathogen circulating and it has to pass through the lymph node, there's a good chance it's going to get attacked. And then your body can mount a defense. Okay, so let's go back to this. So here, we also have something called the spleen. The spleen is on your left-hand side and underneath your ribs, and it acts as a filter for old red blood cells that can be recycled in your spleen. But there's also white blood cells stored here. So, to summarize, your lymph nodes, when you have pathogens circulating in your lymphatic fluid and they flow through the lymph nodes, any white blood cells that live there can go and attack. Versus the spleen. The spleen is where your blood flows through. It's like a net. Think of if you were to go fishing or something, that fine fishing net, or whatever. Like it acts as like a trap. So as the blood flows through the spleen, pathogens can get trapped there, and there's waiting white blood cells to attack or mount a defense. Okay, so, I don't know if you write this. I think you're just thinking about this for a minute. I don't think you write it in a box. Alrighty, so now here, let's look at this cell. Oops. So here is a cell with identification markers on it. So when we learned in the cell membrane chapter, we talked about like identification tags when you wear it around your neck, like, oh look, I'm a Misty Meyer cell. So here on the surface of this cell, there's certain carbohydrate chains that say, hey look, I'm a Misty Meyer cell. Now the cool thing is, all of my cells are going to have the same carbohydrate chains. My blood cells, my skin cells, my liver cells, all of the cells in my body will have the same markers on their surface. It's identical. And, okay, so when we look at the significance of this, if all of my cells have the exact same surface markers, that is one way that my immune system knows that those cells belong to me. It's not going to attack my own cells because all of my cells look the same. Now, when the immune system gets messed up or confused and actually starts to attack yourself, that's autoimmune diseases like lupus, I think MS. And so, yeah, it's kind of like a scary thing when your own immune system is starting to attack you. So now here we have a pathogen. And the pathogen, you can draw in box 7, and then go ahead and add these triangle things to your drawing called antigens. Now, these antigens are a protein or a polysaccharide. So add antigens to your drawing and label it. Now, when we have a pathogen enter into our body, do you think an immune system cell is going to be able to tell the difference between my cells and a pathogen? Yeah, of course, because the carbohydrate chains or the proteins on the surface are different. So as that pathogen circulates throughout my body, I need to have a way to try and defeat it. So we have two types of immune responses that are very specific for pathogens inside of us. So in humoral immunity, we're going to use our B cells. In cellular or cell-mediated immunity, we're going to use T cells. And so both of these systems, or immune responses, humoral or cell-mediated, are going to be used to fight off different pathogens. So in humoral immunity and cell-mediated immunity are our two types of immune response of our third line of defense that we're going to talk about. But first, let's go ahead and talk about B cells and T cells. So each person has about 1 million different B cells. Each person has about 10 million different T cells. And when we look at our B cells, each one is different and unique. So these little Y-shaped things on the surface are the critical components of our immune system. These are what will become antibodies. So when we look at B cells, when this Y-shaped thing is attached to the B cell, we call it an antigen receptor. When they're released, though, we call them antibodies. All right. So during development of B cells, the genes are rearranged because, I mean, to have 1 million different B cells, if you only had, okay, here we go. If you had one gene that led to, like, here's your DNA, led to mRNA, led to a protein, these are proteins right here, you would need 1 million genes just to deal with antibodies. Now, we know that a human has anywhere from 20 to 30,000 genes total, not 1 million. So what happens, how do you get all this wide diversity and variation in antigen receptors is that during B cell development, genes are rearranged and combined in different patterns that results in different mRNA and therefore different proteins. And that's how we can get a wide variety of our antigen receptors. Okay, so that's box 8. Now let's go ahead and look at our humoral response. Now this is for pathogens circulating in the blood. We're going to use B cells for this and the process of clonal selection. So here you have 1 million B cells, and I have different colors to represent the different types of antigen receptors. So with this, you have a pathogen. Now, the pathogen is going to fit very precisely. It's going to match, kind of like we saw with a lock and key in enzymes, but it's going to fit very specifically to one of the antigen receptors. Only one out of the million antigen receptors will fit to that pathogen. So now, once you have a match, this is where the word selection comes from. We've selected which B cell is the match that knows how to defeat this pathogen. So what will happen, once a B cell has been selected, clones are made. Okay, so let's go ahead and just, once we know and identify the B cell, you make a whole bunch of clones. Okay, so therefore, clonal selection. So, okay, here we go. Now for the humoral response. These clones, though, are kind of like your standing army. They're the ones who are going to defeat that pathogen. But the clones actually differentiate into two groups. So we're going to get our clones that are going to be like our memory cells. And they're going to remember this pathogen forever. So when I talked earlier about having chickenpox, chickenpox would be a pathogen that I now have memory B cells for. I will remember, my body has these memory B cells forever. So let's say in 20 years I get exposed to the exact same chickenpox virus that I had in 5th grade. My memory B cells are waiting, and they know how to defeat it, so I don't get full-blown chickenpox again. And then the other type of B cell are effector cells or plasma cells. Sorry, that's cut off by my face, but they're called plasma or plasma B cells. And these are like the snipers of the army. So these plasma B cells are the ones who are going to release their antibodies. And the antibodies will circulate in the blood to go and attach to the pathogens. Now here I only have a few, but in reality they're releasing thousands and thousands per second for their whole life, these plasma B cells. So enormous amounts of antibodies are being released into the bloodstream. So plasma cells will secrete antibodies that are specific for a particular antigen. Okay, so let's go ahead and summarize so far the humoral response and clonal selection in box 9. All right, so now what happens to those antibodies? Well, once the antibodies are circulating in the blood, they're going to attach to those antigens. Now think about this pathogen. If it was a bacteria, could it divide by binary fission and multiply and multiply and multiply? No. If it was a virus, could it get inside of a host cell and start to infect it? No. So now these antibodies have neutralized this pathogen. It can no longer cause harm, and also has marked it for destruction. So as it's circulating in the body, if it goes through the spleen or a lymph node where it finds encounters a macrophage, now it's engulfed and now it's cleaned up. So with that, we have destroyed our pathogen. So here's just an animation showing kind of these little things are your antibodies. You can kind of clump together the pathogen so white blood cells can come and engulf. So with humoral immunity, you can kind of see here that if this pathogen, you can see it's a bacteria with the flagella, has this triangle-shaped antigen, well, there's antibodies specific for that. Where on this bacterium, you can see these antigen receptors will release antibodies that fit perfectly. So our B cells, our lymphocytes, are very specific for the pathogen that they are targeting. Okay. And so with this graph, we can also see the first time you were exposed to a pathogen. This is measuring antibody concentration. So here you have your primary response. It takes some time to build up those antibodies. See antibody here. So eventually, though, once you've defeated the pathogen, that concentration goes down. Let's pretend then a month later, your friend gets the exact same pathogen, and you get exposed to it a second time. Bam! Your memory cells are ready to go. And now your secondary immune response is much quicker, and you don't actually get sick. You might feel off for like a day, but you don't get the full-blown sickness all over again. Okay, so that's your box 10. Now, our next video will be how do you defeat a virus hiding inside of a cell.